Welcome everybody to lecture 7 on blood and hemostasis, or the stopping of bleeding. And in this lecture, I'll primarily be focusing on some of the structure and function of red blood cells, as well as the clotting cascade and the platelets. Okay, to begin, there are some basic characteristics of whole blood that we need to understand. First off, that adults on average have anywhere between 4 to 6 liters of whole blood. All right? And of course, it can range even beyond that, especially depending on the size of the person. Obviously, more blood for a larger person. Now, blood is actually considered a connective tissue type. All right, it is a liquid connective tissue. So if you recall back from your days in undergrad, you might remember that connective tissue comes in various forms, and the hardest connective tissue is bone, and the liquid connective tissue is blood, and most of the connective tissue that we kind of think of falls in between those two, which is more of like a gelatinous type material. In any case, it's a liquid connective tissue, and it does have cells and extracellular matrix, and it's made up of what we call plasma. And so the plasma is the fluid matrix of blood. All right, that's the fluid matrix. And then if we were to separate the plasma from the whole blood, it would look clear and kind of have this like yellow, light yellow looking tinge to it. There are also formed elements which make up the other part of the whole blood. And I call them formed elements. Sometimes it's referred to as the cellular components, but that's not entirely true. I call them formed because they have membranes. These are membrane enclosed structures. So that would be the red blood cells, which are cells, the white blood cells, which are part of our immune system, which I won't be talking so much about today, and platelets. Platelets are not cells, but they are fragments of a much larger cell known as a megakaryocyte. But they do have a plasma membrane, and they do have granules and so on, and they do carry out functions. So we'll talk about that today. Now, another important feature of blood is its viscosity. Generally, viscosity we're talking about is the thickness of blood. How thick is it, right? And it's about 3 times as thick as water. All right, so it's thicker than water. You can think of it as being like thicker consistency, the viscosity meaning kind of almost like a more syrupy consistency, if you will. Not actually as thick as syrup. Okay. In any case, what contributes to that viscosity, what makes it a little bit thicker than water, is primarily things like albumin, which is a protein you guys have heard of, right? That helps maintain our oncotic pressures. And then red blood cells. And so red blood cells are in very high abundance, and I put that in bold because they're really one of the major contributors to blood viscosity. Okay, so you can imagine that clinically speaking, what does viscosity really have to do with the health of the patient? Well, actually it's a lot, because if, for example, the blood becomes too thin, more watery, okay, then it can move through the circulatory system a little bit faster and actually build up into the heart and increase volumes returning to the heart to a greater extent, and it can actually make the heart overwork, and that could in some cases actually lead to heart failure in certain patients, and they usually refer to that as a high output heart failure. On the other hand, the other extreme, we could have blood that's too thick or too syrupy, if you will, where maybe they have too many red blood cells. They're just overproducing it. Maybe it's a tumor in the bone marrow that's actually overproducing it. But there can be various reasons for it. Maybe somebody is taking a drug that's causing red blood cell counts to increase, and that would just cause an accumulation of red blood cells, and that could thicken up the blood and make it slower, make it sludgier. And it could actually, impact the heart as well, because now the heart has to work harder to move this thicker blood, and it could also cause, clots and whatnot. So we'll get more into this as we go along, but that's sort of like an idea of how viscosity plays a role in your health. So the blood plasma. So this is the fluid portion. I'm not talking about the cellular or the formed elements. The plasma has really 3 major categories of proteins. One of which we've already talked about extensively in the capillary lecture, which is the albumins. All right, these are the smallest protein, but they are definitely the most abundant, and so therefore they contribute the most to our oncotic pressure in our vessels. They of course do contribute to viscosity, as I've already mentioned. All right, And there's also globulins. Now, globulins, an example of that would be something like antibodies, which again are produced by our immune cells. And this, of course, provides an immune type function for us. And so they're also in high concentration and do contribute somewhat to the osmolality as well. Fibrinogen, as we're going to learn today, is actually a protein that's involved in clotting. So it helps us to stop blood from leaking out of our vessels when they're damaged. And so that fibrinogen is actually a precursor to fibrin, which will help to form that clot. Now, something I really want to impress on you is sort of the multitude of functions that the liver has. We're going to, hopefully by the end of this semester, really have a good appreciation for what the liver does for us. And then maybe we won't be so hard on it. So the liver, all right, makes these proteins. And the reason I'm pointing it out as sort of an important feature here to remember is that oftentimes in being able to understand how things link together and understand disease better, we have to understand what organs do what. And the liver does so many things. So for example, all right, if most of the plasma proteins are formed by the liver, that includes things like fibrinogen and albumin, for example. So if somebody has liver disease and the function of their liver is starting to fail, that also means whatever it makes is going to be decreased, for example, or maybe even reduced. So they'll have less albumin. Less albumin means that they may not have as much oncotic pressure, so they can start to swell up. So you might see that as sort of the early signs in a patient. You see the swelling could be an early sign of a liver disease, for example. Fibrinogen, it's part of a clotting cascade. So if they have liver failure or liver damage, is reducing its ability to produce these proteins, you might see more bruising and bleeding because it can't form the clots as well. So these are the kind of connections I'm hoping we can kind of make as we move along. In any case, plasma also contains electrolytes. So those are just some of the common electrolytes that you'll see, and of course to varying concentrations. And then nutrients like glucose, you'll see lipids, vitamins, iron, and so on. So there'll be many things that we'll kind of touch on as we move along. So the formed elements, the red blood cells, the white blood cells, and the platelets, they're actually produced from a single cell called a pluripotent stem cell and undergo a process of what we call hematopoiesis. All right, so this is how these cells will actually develop. And so if you take a look at the cartoon I have for you here, all right, you'll see the pluripotent stem cell right there at the top. Okay, and then you'll notice over here if there's certain signals that are involved, certain hormones and certain regulators that will tell the pluripotent stem cell how to divide and what it will end up becoming. And so there could be a group of cells over here, which we refer to as common myeloid progenitor cells. Those common myeloid cells, depending on the stimulus that they receive, can then become any of the following. So over here, what ECFU stands for is erythrocyte. An erythrocyte is a red blood cell, and CFU is a colony-forming unit. So it's just a group of these cells that are now signaled and dividing to become red blood cells. All right, and so the erythrocyte is the red blood cell down here. Of course, these could be other CFUs or other colony-forming units that form, and that could be, megakaryocyte, which ultimately forms platelets. Okay, neutrophils, eosinophils, and basophils, which are all types of white blood cells. All right, and over here, the pluripotent cell could also become a lymphoid progenitor cell. Then it can either become a lymphoblast or a monoblast, and these would form things like monocytes, T lymphocytes, and B lymphocytes. Again, these are all part of our immune system function, so we won't be focusing too much on that for this lecture. But the point here is they all come from that single cell. Due to signaling, they can become, of course, any of those colony-forming units that we just spoke about, and then ultimately become an erythrocyte or become a platelet. They'll actually all form usually from the same part of the body, which I'm going to get into. All right. So vitamin B12, folic acid, iron, things I've kind of highlighted over here. All right. These would be sort of necessary components to producing some of these cells adequately. So as an example, erythrocytes are red blood cells. If there's too few of them in your blood, right, so we have a reduced number of them, we refer to that as anemia. Right? Somebody can be anemic. Now that can be due to a drop in the red blood cell count, or even what we call hemoglobin, which I'll talk about. If there's, let's say, a deficiency of vitamin B12 or folic acid, which is actually vitamin B9, or even iron, the person could become anemic. And you'll learn about different ways to sort of diagnose the host of different types of anemias. Anemia can form from many, many different factors, and I'll kind of mention them as we go along today during this lecture. Okay. All right. So other components and general properties of blood. Erythrocytes, or red blood cells, are the heaviest and settle first. So what we're saying here is if I draw blood, okay, we can take that whole blood and then we can centrifuge it. All right, we're going to spin it around very fast, and when they spin it around, the heaviest components will settle to the bottom of the test tube. All right, so that's what you see here in red. All right, now that component that you're looking at right there is actually the entire red column that you see there are the red blood cells. They're all packed at the bottom. They're no longer mixing around in the plasma. In fact, the plasma has separated out to the top component over here, which is this like beige kind of color. And it's separated by something called a buffy coat, which is right here, which is this very thin white line. So these layers, after we centrifuge that whole blood, we've separated the plasma, or the fluid portion, which is on top, the red blood cells on the bottom, and then that tiny little white buffy coat line is actually the white blood cells and the platelets. And that makes up less than 1% of the whole blood, all right, in terms of, overall concentrations. Now, that means if we were to measure this, we take a look at the ratio of this column of red over this plasma volume here. All right, we take a look at the ratio of that, and it's about 45% of the total volume. Again, the total volume being the entire volume inside this test tube. That column of red there, red blood cells, makes up about 45%, so almost half. All right, and we refer to that as the hematocrit, which is literally just the volume of red blood cells that are in the blood. And for males and females it's a little bit different. Females tend to be a little bit lower, maybe towards like 40%, and males around 45%. The white blood cells make up only about less than 1% of that. All right, and the plasma makes up the rest, the 55 or 60%. All right, and that plasma, of course, is a mixture of the things I talked about. So there's a lot of things dissolved in that water or that plasma. The electrolytes and the nutrients like the lipids and the glucose and the proteins and so on. Now, hematocrit is something we look at in a lab clinically. And when we measure the hematocrit, we see if the hematocrit is normal, which should be about 45%, let's say. If it's less than that, that might indicate an anemia, because it indicates that there's fewer red blood cells, so therefore it would take up a smaller percentage of the total volume. And of course, there are other diagnoses we could make with that in terms of like dehydration and so on. But you guys will get more into that, more to the technicality of those things later on. Okay, so the red blood cell, also known as the erythrocyte, a couple features of it. It has no nucleus, all right, and it has no mitochondria. What that means for the red blood cell is that by not having a nucleus, it can't really make proteins. And if it can't make its own proteins when it needs them, it doesn't repair itself all that well, all right. No mitochondria, so it doesn't undergo any aerobic respiration. It's shaped like a biconcave disc, and I'll show you, a picture of that in the later slides. But that feature of it actually allows it to flow through the vasculature more readily. It doesn't get stuck as easily. It's also flexible due to its cytoskeletal components, and that allows it to kind of bend and contort and snap back into its normal shape again. And that's actually very helpful. Given that it has to kind of squeeze through very tiny capillaries and sometimes can get a little bit distorted. It inside the red blood cell, it carries oxygen on what we call hemoglobin molecules, which is actually one of the features of red blood cells that I'm pretty sure that you guys are familiar with, is that red blood cells carry oxygen to the body, right? So it has to do so on these hemoglobin molecules. And then the last bullet point here is really another feature that some people don't readily think about, which is it actually plays a vital role in acid-base buffering. In other words, we have a pH of our blood which needs to be regulated pretty tightly in order for our cells to function appropriately. And so this will play a role in that, and that role we're actually going to kind of get into more in the respiratory lectures. All right. Over here in the cartoon, what you're looking at is a cartoon of a hemoglobin molecule. All right. The globin portions they're labeling as beta and alpha. These are proteins made up of amino acids. All right. And in sort of the pocket of each of these globin chains here, you can see a heme group, which looks like this red star that's kind of stuck in there. If we were to kind of blow up the red star and look at its actual structure down here, all right, that's a porphyrin ring. They refer to that as a porphyrin ring. And what I want to kind of draw your attention to is, you guys don't need to memorize these structures or anything like that, but this is just to kind of point out a couple of interesting things. At the center of that porphyrin ring is iron. So at the center of each one of these red stars that you're seeing, there's that little gray dot, looks like. That's the iron. So again, this is just to bring back the idea that hemoglobin requires iron. And again, if someone has, let's say, low iron, they might be anemic, right? So an iron deficiency anemia, and because iron plays a role in hemoglobin. And what it does for it is that iron component there helps it to bind to an oxygen molecule. So every hemoglobin molecule can bind up to 4 oxygen molecules. All right, so again, this whole thing I circled right here is one hemoglobin molecule that can bind up to 4 oxygen molecules because it has 4 heme groups that are bound to iron. Okay. Now down here, what you're seeing in this gentleman here is something called jaundice, which you can see in the yellowing of the skin. You can see it in the sclera of the eyes, which is yellow. You can see it underneath the tongue and everything. So what this yellowing is, it can be indicative of a lot of things. There's many diseases that can cause jaundice. All right, but just as an example, if this gentleman had, say, severe liver disease, so we're going back to the liver, one of the liver's jobs is to metabolize and break down old red blood cells, including the heme that's inside the red blood cells, and the porphyrin ring. The porphyrin ring, when it's getting metabolized, turns into something called bilirubin, and bilirubin actually causes that yellowish color. So this is a lot of, say, breakdown of heme causing a lot of bilirubin. If the bilirubin starts to rise in your body and they're not getting excreted appropriately, as the liver would do normally, it can actually build up in the skin there, and you can actually see it as a yellowish color. Now, another example would be, say, if somebody bumped their arm really hard and it started to bruise. Well, it's blood leaking into the tissue there, and the breakdown product of some of the red blood cells that have escaped into that tissue from the bleeding, that yellowish color that can come from bruising, it kind of changes colors a little bit, some yellow and greens and blues and so on, is due to the metabolizing of bilirubin, or of heme into bilirubin. As far as metabolizing of the globin portions, that would really just be broken down into amino acids and redistributed and recycled. The iron can get recycled and so on. But the bilirubin itself, as we'll come back to in the GI lecture, that actually gets utilized and by the liver and dumped into the digestive tract. So we'll talk about its function there later on. So form and function of the red blood cell, I did tell you it has that sort of discoid shape there, that biconcave shape. So it looks like that, all right? So if it were cut in half, you can see the sectional view down here, all right? And so, during development, they actually lose almost all their organelles. So like I said, some of them that they don't have are like mitochondria, nuclei, they don't have them, right? So no protein synthesis, which means it can't undergo mitosis any longer, and it can't readily repair itself. It lacks a mitochondria. So it has to undergo anaerobic fermentation to produce any kind of ATP. This is actually, if you think about it, a good feature to have in red blood cells, because red blood cells carry your oxygen. If they had a lot of mitochondria, they could use up a lot of that oxygen. And this is kind of an example I've given in classes before. If, let's say, you order a pizza, and the pizza guy gets hungry on the way to delivering it to you, and he eats a few slices. Now you're getting half a pie. It's just not as efficient. So the way I look at it is, not having the mitochondria means that he's not going to eat up the pizza on the way to delivering it to the tissue, or eat up the oxygen on the way delivering it to the tissue. In any case, lifespan of the red blood cell on average is about 120 days, at which point they usually get broken down by either the liver or the spleen. and there's macrophages there that'll take care of that. And so they get into the liver or the spleen via those sinusoidal capillaries that we talked about, which have large enough gaps to allow the red blood cells to actually get into the tissue of the liver or spleen. Okay, so one of the major functions of the red blood cell is, of course, transport oxygen to the body. So it picks up oxygen in the lungs, transports it to the tissue so that they can use the oxygen for their energy requirements. And so that means they have to carry a lot of hemoglobin inside their cell to bind to all that oxygen. And in fact, a third of the cytoplasm in a red blood cell is actually just hemoglobin. The hemoglobin concentration is quite high inside a red blood cell. And in fact, specifically, the numbers average about 280 million hemoglobin molecules in one red blood cell. So this thing is like kind of like a sack of hemoglobin, if you will. All right, so yes, so one of its primary jobs simply is to carry hemoglobin, which is bound to oxygen, to deliver to the tissues that require oxygen, right? So again, I shouldn't say again, but the flip side of that is that not only is it delivering the oxygen, but it's also going to help remove waste. Meaning, as the tissues are utilizing all this oxygen, the byproduct of all that metabolizing is going to be to produce things like CO2. CO2 will be taken up by the red blood cells and brought back to the lungs so we can exhale the CO2. And that whole process of dropping off the oxygen to the tissue and taking up the CO2 is something we'll talk about in the respiratory lectures. Last thing, just to kind of impress on you the numbers here, in terms of overall amount of hemoglobin molecules, if there's 280 million hemoglobin molecules in one red blood cell, so that's a lot, right? There are 5 million red blood cells per microliter in our blood. So a microliter is 1/1,000,000th of a liter. So it's a very tiny amount. And there's 5 million red blood cells in a microliter. We have, like I said earlier, 5 liters on average of total blood volume. All right, so you're looking at 5 million per microliter out of 5 liters, and every single red blood cell is carrying 280 million hemoglobin molecules. So if you do the math on that, that's going to come out to well, a lot, right? Okay, so sort of in a normal homeostasis, we would be, producing and destroying, producing new red blood cells and destroying old red blood cells in an equilibrium, so that it can maintain our erythrocyte homeostasis. But as we all know, things change in our environment, and our body has to be able to adapt to it. So our demand for oxygen might go up, and if it goes up for long enough periods of time, well, that might require us to actually make more red blood cells to carry more oxygen, right? And so we have to have a mechanism with which the body can sense and be able to actually tell the appropriate organs to say, hey, we need to start producing more of these red blood cells because we need more oxygen. So the stimulus for increasing red blood cells is something we call erythropoiesis, so making more red blood cells, and we do that with a hormone called erythropoietin. Erythropoietin, which I've underlined for you down here, I just circled it for you. Erythropoietin, sometimes referred to as EPO, EPO. All right. So what happens is this. Let's say I don't have enough oxygen. All right. So I'm feeling, somewhat hypoxemic. So we have here the body is getting hypoxemic. It's not the amount of oxygen we're getting is not meeting the demands of the body. All right. Well, the organs that pick up or can sense that are, surprise, surprise, the liver. Okay, so the liver can sense that, but actually the major one that's going to be doing that sensing here is the kidney. So the kidney can actually sense a drop in oxygen content, and what it'll do is it'll produce erythropoietin and secrete that into the blood as a hormone. Erythropoietin in the blood will actually stimulate the red bone marrow. Now the red bone marrow, as I'll show you on later slides, is actually where the red blood cells are produced. So it stimulates those progenitor cells to start producing more red blood cells. And so we get what we call accelerated erythropoiesis, or production of the red blood cells. We get an increase in the number of our red blood cells. The output starts to increase over time. And so that increases our oxygen transport capability. So we have more cells that can hold on, grab a hold of more oxygen. So our oxygen content will go up, right? So that's the idea behind it, and it's a negative feedback system in the sense that as the hypoxemia declines because of the increase in red blood cells, the release of erythropoietin will go down. And if we start to become hypoxic again, the erythropoietin levels will go up. So this is a negative feedback. Now, there are several things that can stimulate erythropoiesis, and they all kind of circle around various ways to cause hypoxemia. All right, so one classic example is high altitude. So here we're really at sea level here in New York, in Manhattan, Long Island, and so on. But if we were to say travel to Colorado, right, where we're at a higher elevation, where the oxygen content is less, even though our red blood cell content hasn't changed, the amount of oxygen we're bringing in has, and so we'll start to produce more red blood cells. An increase in our exercise. So an increase in exercise demand could stimulate the production of more red blood cells. All right, and in fact, exercising at high altitudes would be a very strong stimulus for the production of more red blood cells. But there's also disease states. For example, emphysema, it's loss of lung tissue. So this is a disease that can be caused by smoking, for example, and this reduces the amount of gases that we can exchange in the lungs. So even if we're at sea level, but if our lungs are damaged and not exchanging the oxygen, what will end up happening is the body will sense that as a reduction in oxygen levels because we're not exchanging it very well, and it'll start to produce more red blood cells. And in fact, that's actually a typical finding in someone with severe lung disease, is you'll see red blood cell numbers are up, and that's in response to the hypoxia that they're undergoing because of their lung damage. Now, the EPO I mentioned, the erythropoietin, it stimulates again those pluripotent stem cells to start to form more of these colony-forming units, these erythrocyte colony-forming units. So it helps to kind of stimulate it in that direction. So it starts to divide, becomes an erythroblast, and then this reticulocyte. And a reticulocyte is a precursor cell, but the reticulocytes will get released into the bloodstream from the bone marrow. And reticulocytes, they're usually less than 1% of the total red blood cell. That's normal. And in about 1 to 2 days, they'll be full erythrocytes, full mature, what we call mature erythrocytes. Now I point out reticulocytes because we can actually measure the amount of reticulocytes in the blood. And again, like I said, it's less than 1% typically. But say if somebody is hypoxic, like someone with lung disease, for example, like I just mentioned, their reticulocyte count might actually be higher than normal simply because they're producing a lot more red blood cells on a normal basis. So it overall increases the percentage of reticulocytes that you could find at a given time in their blood, which will ultimately become the erythrocytes, right? Now down here, what I'm showing you in this cartoon is that red bone marrow produces the red blood cells, okay? Now it does change over time. So in an infant, right here, okay, you can see in their skeleton, their entire skeleton is actually red bone marrow. So they can produce it everywhere from their skull to their extremities, okay? But as we age and go into adulthood, you'll notice that the limbs, particularly upper and lower limbs, they look yellow now because they become more like yellow marrow. They contain more adipose tissue, and they don't undergo any kind of hematopoiesis anymore, or erythropoiesis. So it actually becomes more localized to the axial skeleton as well as the hips. There you can see the hips, and maybe the proximal portions of you can see the femur there and the proximal portion of the humerus there. That would have where all our red marrow is. And in point of fact, like I said before, red blood cells are produced from a common precursor or pluripotent stem cell, which can become any of the lineages, which means in the red marrow, it's not just red blood cells being produced, but the platelets are being produced and white blood cells are being produced. It's important to understand that because if we have any kind of tumor, let's say, or cancer of the bone, that could actually kind of overproduce any of those particular components. So it could overproduce red blood cells and increase the viscosity of the person's blood, right? And that can overwork the heart, etc. As I kind of mentioned before, it can overproduce platelets and increase the amount of clotting that occurs, and so on. If it's the other way around and it's destroying the bone marrow and you're losing the ability to produce these cells, then you can think about how a patient might present. right? So if they don't have red blood cells, they can't really carry oxygen. They might become anemic, right, which make them very fatigued because they don't have the oxygen to carry out their daily activities, let's say. If it affects the platelets, they could start to bleed. If it affects the white blood cells, they might have more infections and so on. And if you see it across the board, anemia, lots of infections, and they're bleeding, well, that should trigger you to think about something going on maybe in the bone marrow, right? Okay, so now on the red blood cells, you guys may have heard of something called these ABO groups, or what we commonly refer to as our blood type. Okay, and so your blood type is genetically determined. Okay, on two different alleles, and so your ABO type is determined based on what we call an antigen on the surface of that red blood cell. And an antigen is really a substance or a molecule that can induce an immune reaction or bind to an antibody to cause an immune reaction to it. Okay, so that's what an antigen is. And we have naturally occurring antigens on our red blood cells. So for example, somebody with type A blood has an A antigen on their red blood cell. Somebody with type B has a B antigen. AB has both antigens, has A antigen and a B antigen. Those are codominant, so they'd have both. Type O, a type O person has neither antigen, doesn't have A or B. All right, so it doesn't have any of those antigens on the surface of the red blood cell. In terms of most common, that would be type O, and the rarest would be type AB. Okay, so as far as the antigens on our blood, you can see over here we have type O, and you'll notice something that actually, if you look at type O, type B, type A, and type AB, all right, you'll see this structure here on the surface. Now, these are actually carbohydrate structures, and you can see the key down here, and you don't need to memorize these galactose, fucose, and so on. You see a chain of them, and the chain is found on all blood types. That's not the antigen. That's not the antigenic portion of it. The antigenic portion of it is the chain, this additional chain. So here on type O, you'll notice there's nothing additional to those three. But on type A, you'll notice this one. It's got this side chain here, this N-acetylgalactosamine. Over here on type B, it's got a side chain of galactose. On type AB, it has both those side chains there. So that's just showing you that it has that sort of carbohydrate branch on the backbone there, and that is antigenic. All right, that can elicit an immune response. And so the antigens are also referred to as agglutinogens, based on the fact that they can agglutinate or clump together when bound to antibodies. All right, they can clump together, and I'll show you what that looks like in the slides coming up. All right, so again, these are glycolipids. All right, glyco is really the sugar portion that I was referring to, and lipid because this whole structure here is bound to the plasma membrane lipid. All right, that's on the surface. We also have antibodies in our blood, and these antibodies, again, coming from our immune system, are called agglutinins, and these are the ones that will bind to an antigen. All right, they're found in the plasma portion, all right, the fluid portion of the blood there, and we have what we call anti-A and anti-B antibodies. All right, so this is just to quickly show how some blood typing can be done, and in fact, you may have done blood typing in a biology lab in undergrad. If not, if you want to know what your blood type is and you don't know, you can actually order a kit online and do it, and within minutes you'll know what it is. And so basically the process is this: you get a kit and you're gonna do a little finger stick, and that's gonna give you a drop of blood, and you put a little drop of blood on one surface, another drop of blood over here. So here, column 1 over here and column 2. So you put down 2 drops of blood, and then what you have is an anti-A and an anti-B. These are the antibodies. So, for example, in the blue vial is anti-A. That means there's an antibody in that solution that will bind to antigen A if it's present on the red blood cell. And you have antibody B in the yellow, and so it'll bind to antigen B if it's present. And what happens when these antibodies bind is they cause red blood cells to clump up. If they clump up and aggregate, we can visually see that, and we refer to that as agglutination. They're agglutinating, or essentially clumping together, because what happens is as the antibody binds to the antigen, it can bind to the antigen on one red blood cell surface as well as binding to another antigen on another red blood cell surface, and kind of hold these red blood cells together. And it can do that to multiple red blood cells, causing this clumping pattern. What we would see is that, let's say, for example, the person has type A blood, and you'll notice that we see clumping. There's agglutination going on, there's clumping, whereas in this spot over here, it still looks like, regular blood. Nothing has happened. So what happens is I've added antibody B on dish 2 there, and no agglutination occurred. So if I add antibody B and no agglutination occurs, that means there's no antigen B on the red blood cell surface for it to agglutinate or bind to. So this person does not have antigen B on the surface of their blood. On the other hand, if I add antibody A and it agglutinated, which you can see here, this is the little clumping that you're seeing here. In that case, that means that antibody A bound to antigen A on that person's blood. So therefore, this person is type A. Type B, you see it's the reverse. The antibody B agglutinated the blood. So therefore, antigen B was located on their surface, so this person has type B. It didn't do anything here. Type AB, you notice in both cases it agglutinated, so both antibody A and antibody B. So therefore, this person's type is AB. And then last, of course, O, which doesn't have any of those antigens. These antibodies won't work. Neither A or B would work, so therefore there's no agglutination at all, and so they're type O. Okay, so this is just a visual to help you see how the antibodies are working. So in the previous slide, I was talking about using it to help test your blood type. We artificially give antibodies and put them on the dish to see if it reacts with the person's blood, and if it does, then whatever that antibody was indicates the type of blood the person has. So if antibody B causes agglutination, then they have blood type B. So here are the antibodies here in green, and so it can bind to the antigens on the surface. So in this case, if it's binding to the antigen on the surface of the red blood cell, you'll notice it has several of these little Y arms on them. Well, they can bind to several different antigens, which means they can actually clump these red blood cells together, as you kind of see here. As each one of these is binding to different red blood cells. This can form massive clumps, what we refer to as agglutination, and we can see that visually, as you saw in the previous slide. The problem with that inside our bodies, if this is happening, and the most common scenario for that to happen, is when we're getting blood transfusion. So if somebody receives blood, and let's say their blood type is type B, and they get transfused type A. What happens is our bodies naturally, if you're a type A blood person, let's say, you would naturally make an antibody to B. So you make an antibody for the antigen you don't have. So again, if I'm, let's say, type A blood, I would have antibody B present in my blood. So let's say I give a person with type A blood, I give them, or I transfuse them, type B blood. What's going to happen as soon as they get that type B blood, that means that blood that they just received has the B antigen on it. I already make antibody B. I have the antibody for the type, for the type I don't have. So it's going to bind to that red blood cell and react to it and cause that donor blood to clot or form these, these clumps. The problem with that is that we can get what we call a transfusion reaction. That can actually, those clumps of red blood cells that are clumping together due to the antibodies binding to it, can block small blood vessels. It can also, weaken the red blood cell membrane, cause hemolysis or rupture of the red blood cell, and that can release hemoglobin. And tons of hemoglobin pouring into the vasculature there, into the plasma. Well, that can actually do damage to tissue, particularly the kidneys, for example. So here, hemoglobin can block the kidney tubules and cause acute renal failure. There are various degrees of transfusion reactions, anything from kidney failure to causing strokes to death. You could also have mild allergic reactions. So there are sort of a wide range of reactions that can occur. So as an example of that transfusion reaction, right, if I get agglutination because I mismatched the blood type, you can see that there's clumping here of red blood cells, and as they travel, they could get wedged in some of these smaller branched arteries for example, or veins, and that could end up having, very very poor effects on the body, right? So here they're saying, this is type B, so they have anti-A, and this is the recipient. So this person has type B blood, which means they have antibody A. The donor red blood cells are agglutinated, which means we mismatched it. So probably, let's say they got type A blood, and so their antibody A's are attacking these type A blood. It's causing this agglutination. All right, so let's try out this chart for a second. So let's see if they had type A blood. This person has type A blood, which means on the surface of their red blood cell, they have antigen A, okay, as we've gone through, which means that they produce antibody B. So in their plasma, they would naturally have antibody B. All right. Now, interestingly, we actually aren't born with those antibodies. We develop them very quickly, usually within weeks to months after we're born, from our normal gut flora. So they can kind of invade the blood, and so apparently it mimics some of the antigens that we see on red blood cells. And so typically, based on that, we will develop the antibody Bs, after birth, after we're exposed to our gut flora. So in any case, someone with type A blood would have antibodies to type B. So the question is, all right, well, who could they receive from then? Well, they could receive blood from somebody with A, so they could receive type A blood, right? They could also receive type O blood. All right. The reason they can receive type O, let's take a look at type O blood. Somebody with type O has no antigens, so there's no antibody to it. It wouldn't cause an antibody reaction, so they can receive O. Who could type A donate to? Well, they could donate to anybody who has an A antigen, so that would be type A, right, of course, and type AB. Because person with type AB, let's take a look at that. Why could they receive it? Well, they're type AB, which means they have antigen A and B. They don't make any antibodies. So somebody with type AB blood doesn't produce any antibodies, therefore you can safely give them type A. You can also safely give them type B, so I'll put that in there. And they could also receive B. So B, if you have type B blood, your antigen is B. You have antibody A. Again, the antibody to the type you don't have. So they could receive from B or O. Okay? And can donate to B or AB. Now, if you have type AB blood, that means you have antigens A and B. And so therefore, you don't make any antibodies. Okay, you don't make any antibodies at all. So in that case, since you don't make any antibodies, you can receive A blood, B blood, O blood, or even AB blood. So you could receive from anybody. So that's one of the benefits of having A blood, is the fact that AB blood is that you don't have any antibodies. So therefore, you won't react to any of those types of blood. So we refer to AB, all right, as the universal recipient. They could take any type of blood. Now, on the other hand, in terms of who you can donate that to, that's a little bit different. You could donate them to AB. All right, so we can donate to AB because, we can donate to AB only. You couldn't give it to somebody with type A blood because it has a B antigen on it, and their antibody B would bind to it and cause hemolysis, right? Now, type O, all right, there are no antigens, and because there's no antigens on the surface, that person produces both anti-A and anti-B, which means for O, they can only receive from other O. All right, they can only receive from other O because that's the only one that doesn't have any other antigens. Because otherwise, you give them A, B, or AB, they have antibodies for all of those, right? However, people with O-type blood can donate to A, B, AB, and O. Since it doesn't carry any antigens when you give it to somebody, if they're AB, or O, all right, they won't cause any kind of reaction. So in this case, O is considered the universal donor. So AB is the universal recipient, O is the universal donor. And you'll notice that we've given O in every case here. You'll notice that we can donate to AB in all cases there. So again, AB, universal recipient, O, universal donor. All right. All right. Now you may be asking yourself, all right, well, if I give somebody O-type blood, mixing that O-type blood will also be the antibodies that they have, like anti-A and anti-B. What about those antibodies? Would that react with the host blood? So, for example, if I give type O blood to somebody who's AB, when I give type O blood, it's going to carry with it the antibodies A and B, and won't that hurt the host's red blood cells? And so that can be minimal, but what can be done to kind of avoid any of that kind of interaction is sometimes you can give donor plasma that may have just like more like donor blood that might just have packed cells in it and less plasma to avoid the antibodies that might be dissolved in the plasma. And they can also use other techniques to actually help to eliminate some of the antibodies in there. So there might be some processing that goes on of the donor blood to remove some of those antibodies. All right, so now I want to talk about the Rh group. And the Rh group is actually something referred to as the rhesus factor or rhesus antigen. And so there are multiple types, C, D, and E here, for example. And this antigen is actually denoted on our blood type as a positive or a negative. So you might be thinking, we talked about the ABO groups, but what about the A positives or the B negatives? Well, that positive or negative is actually that Rh group. Specifically, the RhD, meaning it's the RhD antigen. This D antigen is the most reactive and is considered part of your blood typing. So it's something we look for, all right, since it can cause a reaction. And so, for example, if somebody is A positive, that means they have the A antigen as well as the D antigen on their blood on the surface of the red blood cell. Now, if I say somebody is A negative, that means they have the A antigen and they lack the D antigen. They do not have it on their red blood cell. That's what that means. Now, if you recall from the previous slides, if somebody has blood type A, that means they produce antibody B, right? However, in somebody who is negative, Rh negative, they don't produce an antibody D. Right? They don't produce an antibody D to the D antigen. It's not normally present. I'm going to underline that here. It's not normally present. However, under certain circumstances, it can become present. Right? So it doesn't naturally occur like it does like having antibody B or antibody A. All right. So the way it kind of occurs is because we get exposed to Rh positive blood. So if somebody has negative type blood and they're exposed to the D antigen, all right, from somebody else's blood, that can actually cause the development of the antibody. So here, as an example, let's say this is one of the most common scenarios, is in pregnant women, where you have a woman who actually has negative blood, Rh negative blood. So they're B negative, A negative, and so on, right? But the fetus is positive. So they might be A positive or B positive and so on. And so what can actually happen in that scenario is that some of the blood from the mother and the fetus can actually be exposed to each other, all right? Because there can be some bleeding and some transfer of fluid between the mother and the fetus, and in those cases, that's where the mother's blood will actually be exposed to that positive or that D antigen, and she can start to produce antibodies. So we'll talk about more of that in a second. The other common way is from transfusions. So somebody who has to receive a lot of transfusions, yes, they are at higher risk for being exposed to positive blood and kind of over time develop that antibody. All right, and in general, the rule of thumb here though is that there's no problems with the first transfusion or pregnancy when they're first exposed to it. There's usually no reaction at all because it takes time for that antibody to develop. Once the person has developed the antibody, though, once they're exposed to that positive blood again, then they can form a very severe reaction. So in the pregnant woman, you have what we call hemolytic disease of the newborn. All right, this again occurs if the mother is Rh negative, all right, and she's already formed the antibodies. So, for example, she might have, say, been Rh negative and her fetus was Rh positive in the first pregnancy. There was some exposure. It didn't hurt the first pregnancy because the antibodies hadn't had enough time to form yet. However, if she gets pregnant again, if she has another fetus that's Rh positive, now she has these antibodies, and those antibodies can cross the placenta freely. If they cross the placenta, they can actually attack the fetus's blood cells. If they attack the fetus's blood cell, of course, that can result in death of the fetus. Okay. So this is why we say with the second child is at risk, not the first. All right. Now, in terms of prevention of that, of course, there's something called RhoGAM, which is given to a pregnant woman who is negative. So if she's A negative, B negative, and so on, you would give them RhoGAM at a certain time. And the RhoGAM actually helps to kind of disguise, if you will, or hide the antigens from the immune system of the mother, so she doesn't actually produce them in case she gets exposed to the fetus's blood. As you see here in the hemolytic disease of the newborn, you can see the fetus there, and there's some communication initially between the mother and the fetus's blood. So this is the first pregnancy. There's no harm to the fetus in the first pregnancy, but you'll notice here she's developed the antibodies. So that's what those little green Y's are. All right, so this is this anti-D antibody, and now she has a second child, and you can see here in that second pregnancy again the fetus is Rh positive. And that's going to result in these antibodies that have been formed crossing the placenta and attacking the fetus and hemolyzing their blood and causing clotting and so on. It could result in the death of the fetus. Okay, so now I kind of want to change gears a little bit and talk about hemostasis. And this is, the stopping of blood. And obviously this is a very important mechanism for our homeostasis, because we don't want to lose too much volume, right? If we have damaged blood vessels, and this occurs readily throughout our day, we have lots of nicks and cuts and bruises and so on. But in fact, the endothelium that lines our blood vessels is under a lot of stress on a daily basis, and so they do get damaged quite frequently. And we have to have mechanisms in place to make sure that we can stop bleeding quickly and effectively. All right. And so we need to form those clots. So we're going to go from basically a fluid medium in the blood, and then locally turn that fluid into something solid that precipitates out of that solution and forms basically a solid clot, and then stays there for a long enough period of time to allow the tissue to heal. Okay. Of course, there's abnormalities of the system, and that can result in too much bleeding, or the flip side of that would be too much clotting. Okay, and I'll touch on that in a little bit. But there's two components. We have the platelets, which are also referred to as thrombocytes, and we have coagulation factors. All right, so let's talk about the stages of hemostasis. So there's three, and the first one is vasoconstriction, or what they'll refer to as vessel spasm, and this is really just a temporary step, or a very transient step, and part of it's thought that this helps to reduce blood flow to that region in order to reduce the amount of blood lost through the damaged site. Then there's step 2, which is the formation of the platelet plug. So we're going to activate platelets, and they're going to be sort of the initial responders. And then step 3 is blood coagulation. And the ultimate goal of blood coagulation is to form what we call a fibrin clot. Okay. Now platelets play an important role in all three. All right. So in fact, platelets sort of being the initial responders will actually initiate vessel spasm. They can obviously form the plug, but they'll also play a role in blood coagulation, and blood coagulation can also play a role in recruiting more platelets. So they're kind of all intertwined together. And ultimately the goal is to form a thrombus. And a thrombus just means we're forming a clot. And now a thrombus may sound bad in terms of, when somebody has a thrombus, we think of, certain diseases that occlude vessels and so on. But actually a thrombus, physiologically speaking, is perfectly normal as long as the mechanisms that control it are working appropriately. And so it can maintain that clot to the injured site and not in areas where we don't want it. So let's talk about the platelets. All right, these are thrombocytes. Compared to the red blood cells, they don't live very long. I mean, it's 120 days for red blood cell. It's 10 days for platelets. They are anuclear as well, because these are fragments of megakaryocytes. They're not able to synthesize new proteins. Okay. They contain a protein, a very important one called cyclooxygenase. And cyclooxygenase is important for producing an eicosanoid that I'm going to be talking about in a few minutes. They contain microfibrils. All right, so these help to promote the release of granule content when they become activated. All right, so there's a host of intracellular reactions that are going to occur. They also contain what we call an open canalicular system. And that's in the cartoon down here, that's the open canalicular system, like channels or what have you. But you'll notice this is actually the platelet, when it's not activated, is spherical. And it contains a whole bunch of granules, all right? And then the granules themselves over here are referred to as dense granules. Now what you can see here, dense and alpha granules, these alpha ones. What's in the dense granules are ADP, all right, ATP, calcium, and serotonin. This is all going to play a role in this cascade I'm going to talk about. Alpha contains proteins like von Willebrand's factor and coagulation factors 5 and 13. So this is like the first instance where we can see platelets actually contain coagulation factors. Factors are these little proteins that are part of the coagulation cascade. All right, so they're releasing this to help promote the coagulation cascade. Inside the cell, they also contain actin and myosin, which forms pseudopods, which just means false feet, or just these sort of extensions out from the body. And thrombopoietin induces platelet production. Just like erythropoietin produces red blood cell production, thrombopoietin induces platelet production. And as you guys know, that's in the red bone marrow. So if we look at the cartoon, the platelets have this sort of spherical shape. They contain those granules, all right? And then when they're activated, I'll draw the arrow here, when they become activated, they can take on this sort of star shape. And what ends up happening is, on the outer edge, if you look at the normal platelet for a second, the outer edge, you can see kind of what looks like brown lines. Those are really the microfibrils that help give the platelet its shape. And they actually will start to kind of, in a motor sense, like muscle contraction almost, they start to move inward. And it starts to group the granules all together, the dense and the alpha, into a very dense spot there, and it increases its surface area with these extensions. All right, it's what we call these pseudopods. So it increases its surface area. So that's part of the activation. It changes its shape, sends out these pseudopods, increases its surface area, and it's going to degranulate. It's going to start to release the contents of its granules with this new increased surface area, allowing it to really dispense of all that. But that increased surface area is also going to be revealing receptors that weren't revealed when it wasn't active. And this will allow it to bind more readily to structures around it. Okay, so that first step in hemostasis was vasoconstriction. Now, from an activated platelet, it's going to release thromboxane A2, TXA2. All right, so thromboxane A2. This is the eicosanoid I was referring to on the previous slide. Cyclooxygenase is a protein located in the platelet, all right, that acts as an enzyme in the pathway that produces thromboxane A2 from the plasma membrane. Thromboxane A2 gets released along with serotonin from the granules, and both serotonin, which is also referred to as 5-HT, these two can actually both stimulate vasoconstriction. All right. Now you can see this in large and small blood vessels. All right. It'll reduce the blood flow, but it doesn't stop the blood flow because we still need blood flow to get other platelets there. We need to start recruiting some more platelets to fill in and plug it up. So we don't want to occlude the vessel. We just want to slow it down. Now to form the plug at the damaged site, this is how it works. If, let's say, in one of the blood vessels, I've damaged the endothelium. Remember, the endothelium is what lines the inside of the lumen of the blood vessel. Okay, and if there's damage, the endothelium is disrupted and it exposes the connective tissue underneath it. And one of the primary components of that connective tissue is collagen. Collagen has an overall negative charge to it. And that exposed collagen with its negative charge stimulates and activates platelets that are nearby. So the platelet being exposed to the collagen becomes activated. Once it's activated, I need several things to happen. I need, first off, I want to plug up that hole, that damage now, because I don't want the blood or the plasma getting out. So the platelets have to, when they activate, bind to each other. In other words, we refer to that as aggregation. When one platelet combines with another platelet, they start to aggregate. I also need that aggregate of platelets to stay at the damaged site. So I need it to adhere to the damaged site. So aggregate and adhere. So in that way, I've got basically what we call our platelet plug, which is staying at the damaged site. So like I said, the initial exposure to that negative charge of that subendothelial collagen, that'll activate any nearby platelets. Once those platelets that are activated by that collagen exposure, they'll start to change the shape that you saw on the previous slide and degranulate. They'll start to release thromboxane A2 and serotonin, for example, to cause that vasoconstriction. And the platelets will also release ADP along with our thromboxane A2. So in this case, ADP and thromboxane A2, they actually get released into the blood, and any platelets that are going by that haven't been activated yet will become activated in response to ADP and thromboxane A2. So thromboxane A2 actually has two roles: vasoconstriction with serotonin, as well as recruiting or activating more platelets along with ADP here. So now they're all becoming activated, and this is a positive feedback, because what's happening is once you activated those platelets, they're also going to degranulate, and they're going to release ADP and thromboxane A2 and serotonin. So there's this positive feedback loop that's occurring. Okay, and this is what we want, because we want more and more platelets to become activated and adhere to that site to form a nice solid platelet plug. So now, in order for them to aggregate and adhere, these activated platelets will expose proteins on their surface when they become activated. One of those proteins is glycoprotein 2B3A. And I know it's not a great name, and these names are really pretty terrible, but we have to remember them. Glycoprotein 2B3A. What it does is, when it's exposed, it binds to fibrinogen. So fibrinogen is in our blood plasma. It's just, it's dissolved in it. So what will happen is it'll bind to it, and fibrin is actually this long filamentous protein. It'll bind to that exposed glycoprotein 2B3A, and it'll link the platelets together because they're exposed on multiple surfaces of multiple platelets, and so they can actually bind to the same fibrinogen molecule, all right, or protein there. And so they bind at various sites, and as you can see here, by linking to this one fibrinogen protein strand, we have a couple platelets linked to it. And so they can do that throughout, and they can cross-link them and whatnot, so they can become aggregated together. So that's good. So we have an aggregate. Now, how to adhere is based on another receptor that gets exposed, and that's the glycoprotein 1B95. Horrible name. But anyway, I'll just refer to that as glycoprotein 1B for now, all right? So this glycoprotein 1B gets exposed. It responds, or it binds to, von Willebrand's factor. Now, von Willebrand's factor, this is another protein that actually binds to the collagen. So von Willebrand's can actually bind to the collagen. It's located in the plasma, as well as being released from platelets. So von Willebrand's, once it's released and it's also in the plasma, can bind to that damaged surface, bind to the collagen, and then the glycoprotein 1B on the platelet surface will bind to that von Willebrand's. So von Willebrand's acts as a bridge between the damaged site and the platelet, and the fibrinogen acts as the bridge between platelets. So now we have our aggregate, and they can adhere. So they adhere because of von Willebrand's and glycoprotein 1B. So this is a little bit of a summary here. All right, thromboxane A2 and serotonin, or 5-HT, causes vasoconstriction when it's released from the platelet. Thromboxane A2 and ADP will promote activation of the platelets, and by promoting activation of the platelets, it'll also allow the platelets to expose those glycoprotein receptors to bind to fibrinogen and von Willebrand, so they can aggregate and adhere. So that's what the von Willebrand here and fibrinogen are for. All right, and then the platelet phospholipid membrane. So the membrane of the platelet itself is actually going to be a site for coagulation to take place. So this is again where platelets and the coagulation cascade are kind of go hand in hand. They need the phospholipid membrane of the platelet to carry out some of the function of the coagulation cascade. And in the drawing down here, what you're seeing in the leg there is it's not a rash. Those are actually what we refer to as petechiae. And so these are actually small ruptured blood vessels. And you can see these are essentially micro bruises, if you want to look at it that way. These are like little bruises throughout, or little hematomas, all right? And they're like almost pin size, some of them. And this can happen in certain abnormal conditions where there's too few platelets. And this is just to remark on how often this is going on in our bodies. What we don't realize is that in a 24-hour span, we can have thousands of little nicks or damages to our endothelium. Thousands, because they happen at joints where we're constantly compressing them and re-expanding them. They can tear, and they do it frequently. And they're just tiny microscopic tears, and sometimes only in very small vessels. And our platelets take care of that. They'll take care of thousands of them a day. We don't realize how often this is actually occurring because of the efficiency and the effectiveness of this system, until you have diseases where, in what we call thrombocytopenia, which is just a lack of platelets, can result in what looks like a rash, but is actually tiny little bleeds caused by the lack of platelets to be able to plug up those small holes. All right, so this is to demonstrate to you, how often this is actually occurring and can happen throughout our bodies. So in this cartoon, what you're seeing here, all right, is the damaged site right here. Okay, and in that damaged site, you'll see that there's exposed collagen, which is the green. You see that here and here. So that has that negative charge. That's going to activate the platelet. The platelet's going to change its shape once it becomes activated and degranulate, expose some of those receptors. And so you see the change in shape occurring. All right, it's going to release contents like ADP and thromboxane to recruit more platelets that are coming by. So we get that positive feedback. And of course, serotonin, thromboxane, A2 would cause some vasoconstriction, but the serotonin is not listed on here. All right, but the ADP and the thromboxane A2 are responsible for the recruitment of those platelets. And you'll notice that they adhere and aggregate to each other, and that's again because of the exposure of the glycoproteins 1B and 2B3A in the presence of fibrinogen and von Willebrand's factor. You'll notice in the normal endothelium, both upstream and downstream of the damaged site, all right, platelets are not being activated there because of the healthy endothelium prevents that by releasing things like prostacyclin, all right, and nitric oxide. Prostacyclin and nitric oxide actually inhibit platelet aggregation, and that helps to keep any platelet activation from occurring abnormally or any kind of aggregates forming in regions that are not by the damaged site. All right, so now we're going to talk about coagulation, which is the third step in the hemostasis process, and this can actually occur simultaneously while the platelet plug is forming. In coagulation, this is essentially clotting, and it's the last and most effective defense against bleeding. And so the ultimate goal here is to take a bunch of inactive proteins to activate them in a cascade that kind of has like a domino effect, that ultimately the end goal is it's going to lead to the activation of fibrinogen. So it's going to convert the inactive fibrinogen into this fibrin, okay, and this fibrin becomes insoluble, and it can actually cross-link with other fibrin monomers, and it creates sort of like a mesh or a net that will actually cover the surface of the platelet plug, adhere to it, and actually help to reinforce it. The procoagulants, or otherwise known as the clotting factors, are basically the cascading proteins that go from an inactive state to an active state, and the active ones will then activate some other protein and so on. So it's like a cascade of activations. And there are actually two pathways that these clotting factors can take. So it's what we call the extrinsic pathway or the intrinsic pathway. And so to initiate the extrinsic pathway, the blood has to be exposed to the tissue surrounding the blood vessel. So we have to damage the blood vessel so that the external tissue is exposed. And the damaged tissue that surrounds that blood vessel releases what we call tissue factor, which is actually factor 3. That factor can actually then initiate the extrinsic pathway and its related factors until it leads to the formation of fibrin. Now, the intrinsic pathway, based on its name, actually takes place, the damage takes place entirely within the intravasculature. So that means that it could be the endothelium that's damaged, that lines the blood vessel, or the subendothelial collagen, which is just underneath the endothelium. So that exposure could also activate the intrinsic pathway. There are some other initiators of the intrinsic pathway as well. Certain inflammatory mediators in the blood could also do it. Now, the reality is we have these two pathways, but they have crosstalk between them. So they'll actually, in reality, any kind of damage, let's say we had tissue damage, when we activate the extrinsic pathway, we'll also be activating the intrinsic pathway as well. There will be certain circumstances and pathological conditions where you might activate one pathway primarily over the other. So, for example, if somebody has atherosclerosis, all right, which basically you have, these deposits that are occurring in the subendothelial space, that can damage the endothelium, and that could lead to activation of the intrinsic pathway more prominently than, say, the extrinsic. But again, since there's crosstalk, the reality is usually those pathways will converge and they'll kind of reinforce each other and produce that fibrin. So regardless of the pathway that's initiated, the end goal or the end product is the same, which is production of fibrin. So yes, for this lecture, or for the exam, you'll have to know the pathways and all those factors. Okay. Now, some of the factors are given, specific names, and I won't expect you guys to know that, but you will need to know the factor numbers. So you may have to review your Roman numerals. But a couple things before I start that: calcium. All right, calcium is required for either pathway. So calcium is actually very important for clotting. And in fact, clinically, if they draw blood, sometimes they'll actually add a chelator of calcium, something that helps to remove the calcium from the person's blood so it doesn't clot. So calcium is absolutely required for both pathways, intrinsic versus extrinsic. The vitamin K dependent factors, we need to know what those are. So those are going to be actually 2, 7, 9, and 10. We need to know that because when you anticoagulate somebody, where we heard of the drug warfarin or Coumadin, for example, when we want to reduce their likelihood of clotting or forming a clot, because there are certain pathological conditions patients can develop where they're at risk for clots, things like warfarin or Coumadin, they actually inhibit vitamin K, which reduces those clotting factors, 2, 7, 9, and 10, and reduces their likelihood of forming that clot. Most factors, like I mentioned, are produced in the liver. In the cartoon, which you can see over here to the right, we have the intrinsic pathway in blue. You can see in pink over here is the extrinsic pathway, and you notice the extrinsic pathway is much shorter. So look at the intrinsic. Intrinsic is also referred to as the surface contact pathway because it takes place essentially internally inside the blood vessels, so it's the surface of the blood vessel, the endothelium. And when that gets damaged, we activate factor 12 into 12A. So the little subscript A following the Roman numeral means it's activated. Then that can convert factor 11 into 11A. 11A will act on 9 and convert that to 9A. Once we have 9A, 9A is actually going to combine with factor 8. PL stands for the platelet, for the phospholipid. So that's the phospholipid membrane of the platelet. So in point of fact, a lot of the reactions where these different factors are interacting with each other actually occur on the surface of the platelets themselves. So the platelets act as sort of the grounds for where these coagulation factors can interact with one another and is required and necessary. Here's some calcium, which you can see in parentheses there. So this complex of 9A, 8, the phospholipid plus the calcium creates a complex that's going to convert factor 10 into 10A. Once we hit factor 10, however, what we'll call the common pathway. We call it common because if we look at the extrinsic pathway, which is really short, that starts with the tissue factor, which is released from the damaged tissue, which is factor 3. And that's going to interact with factor 7, 7A there. All right. And the complex between tissue factor and factor 7A is going to activate 10. So the intrinsic and extrinsic pathway, they funnel down to activating factor 10. So 10 is the start of the common pathway. Extrinsic pathway is much shorter and it's actually faster. Okay. And the common pathway starts with factor 10. So once we have factor 10A is going to complex with factor 5, phospholipid, and calcium, which you can see there in parentheses. So that complex of 10A, factor 5, phospholipid, and calcium is going to activate or turn prothrombin into thrombin, and thrombin is a major player here. All right, so thrombin, prothrombin, and thrombin, those are factor 2. All right, so thrombin is factor 2. Thrombin is going to convert fibrinogen right here into fibrin. Fibrin is factor 1. And then finally, once we form this fibrin, it'll cross-link with each other, but that's more of an ionic bond and not as strong. So factor 13 over here, factor 13's job is to actually cross-link the fibrin monomers with each other so that they're covalently bound, and it strengthens their bond. So factor 13 helps to cross-link the fibrin, and it ultimately forms a stable fibrin clot that surrounds that platelet plug. And you'll see here that 13 is activated by thrombin. All right, so thrombin not only converts fibrinogen into fibrin, but it activates 13 into 13A, which is going to help cross-link that fibrin into a nice, stable, strong mesh. So this whole thing is like a series of dominoes, which I kind of show the picture there, where it's a chain reaction of activations. Now, as I mentioned in the slide previously, the extrinsic and intrinsic do crosstalk with each other, and there is feedback loop. So this is all basically a positive feedback mechanism that's going on here. So as we start to produce downstream mediators, like for instance, as more 10A is being produced, 10A is going to act back on the pathways of the extrinsic and intrinsic pathways to enhance the activation of some of those factors to amplify its own production. Thrombin can do the same thing. So this is all sort of enhance their production and will produce lots and lots of fibrin to create a nice sturdy mesh in a very quick, do it very quickly. Okay, so what I'm going to do here is draw out a little overview of the cladding cascade and a little bit of a review here. So let's say there's an injury to the blood vessel. Over here I'm going to draw our initial response, which was the platelet activation. We have platelet activation response to that exposed collagen. And what it's going to do is it's going to release a couple things, right? We're going to release ADP, also going to release thromboxane A2. It's going to release serotonin, 5-HT, right? And so thromboxane A2 and 5-HT are going to cause vasoconstriction. Thromboxane A2 and ADP are going to activate more platelets. All right, I'm sorry for writing so small. I'm doing the best I can to fit all this in here. So that's platelet activation from the injury. All right. Again, it's going to release some von Willebrand's factor, and in the presence of some fibrinogen, those platelets that are being activated, all right, the platelets are going to adhere as well as aggregate to the site. Aggregate to the damaged site, and that's going to form my platelet plug, which was my step 1. All right. Now let's say I've activated the extrinsic pathway, so I've damaged it, I've damaged tissue, and damaged tissue releases tissue factor. Tissue factor, which is factor 3, is going to initiate the extrinsic pathway. So extrinsic pathway. All right, again, this is because of tissue damage. All right. So that damaged tissue is going to release tissue factor. So tissue factor, or TF, which is actually our Roman numeral factor 3. And that, in the presence of calcium, and in the presence of some of the spontaneously activated factor 7, is going to result in a complex of tissue factor plus 7A, the little a, again, denotes that it's activated, plus our cofactor calcium. All right, so it's going to form a complex that will then turn factor 10 into factor 10A. So we'll activate 10 into 10A. Now factor 10A, in the presence of calcium, plus factor 5, and all right, the plasma membrane. So I wrote plasma membrane here to let you know that this reaction is taking place on the plasma membrane or the surface of a platelet. So this is an entire complex that's now going to form (10A) combined with calcium combined with factor 5 on the plasma membrane. Now again, this is 5A, excuse me, so 5A. We refer to this entire complex here as a prothrombinase complex. So factor 10, factor 5, calcium in the plasma membrane form the prothrombinase complex, which means that this complex is going to convert prothrombin, I'm going to draw the arrow over here, prothrombin into thrombin. Now I'm going to put a little block around thrombin because it's an important intermediate here. Thrombin is going to convert fibrinogen into fibrin. And as you know, fibrin is the last step here, which is going to help us form that mesh to really cross-link and lay over that platelet plug. So the platelet plug plus our fibrin mesh are going to be what we call a thrombus. Now, fibrin alone is not going to be enough in order for us to form that nice strong mesh. In fact, it needs to be cross-linked by another factor. And that factor is factor 13. So factor 13's job is actually to cross-link the fibrin monomers to each other. What that really means is it turns what's essentially an ionic bond into a covalent bond, which would be much stronger. And so it's cross-links them, turns them into covalent bonds, so create a nice tight mesh that goes over the platelet plug, and we have our thrombus, or our clot. And that's through the extrinsic pathway. If, on the other hand, we are activating the intrinsic, I'll draw that over here. The intrinsic. So in this case, with the intrinsic pathway, what's happening is the blood has been exposed maybe to just the collagen, or there's some sort of internal blood trauma, maybe due to the immune system and some of the intermediates of the an inflammatory reaction. And so what happens is we'll expose that, let's say that collagen, and we'll activate 12 into 12A. 12A can then convert 11 into 11A. 11A can then convert 9 into 9A. And then 9A in the presence of calcium, of course, as well as factor 8. We want factor 8A. So that's going to combine here along with this and this. So these are all combined. 9A combined with calcium and factor 8A. And what they're going to do is they're going to be able to turn factor 10 into factor 10A. So they converge on what we call the common path. So we reach the common pathway as soon as we reach factor 10. Once we activate factor 10, we've reached the common pathway. So what happens now is, like, under normal circumstances, if we had an injury, both the extrinsic and intrinsic pathways would typically interact with each other and both be activated. We'd also get platelet activation, and then there's interaction between the platelets and the coagulation cascade. Now, under normal circumstances, as we activate, okay, this feedback loop, this positive feedback loop, is enough to cause some of the clotting, but it's not enough. We actually have to amplify these coagulation cascades. We have to amplify the whole process to really kind of to rev it up to clot very quickly. And so what's going to happen is there's a few factors that play key roles here. So I'm going to use the color purple to kind of identify them. So we have factor 10A, which I'll put a circle around in purple. So factor 10A is an important intermediate because what it does is when it becomes formed, not only does it become part of the prothrombinase complex, but some of it will actually go back and activate more factor 7. By activating more factor 7, I increase the proportion of activated factor 7, which can bind more tissue factor, which is being released in abundance. So it's promoting its own formation because by activating more 7, it can bind to more tissue factor, and that can ultimately form more activated 10. So I have a small positive feedback loop here, which really helps to amplify the process. Now on top of that, factor 7 can actually activate factor 9. So this is where there's crosstalk between extrinsic and intrinsic. Factor 7, since that's getting amplified as well, can activate more 9. More amplified 9 means binding to calcium, and 8 will result in more 10A. So it's once again 10. Factor 10 is amplifying itself. Okay. And now another key here is thrombin, which I put a box around here. So thrombin can actually activate platelets itself. So it can actually act to increase platelet activation. And it plays a key role here by activating factor 5 and activating factor 8. Now, for the most part in these cascades, for example, if I activate 12, 12 can activate factor 11 and so on. They act as enzymes that help to kind of turn their inactive form into active forms. However, factor 8 and 5 are a little unique in the sense that they're actually more like cofactors. And a cofactor is something that can help enhance or amplify enzymatic activity. So maybe on the first path or the first run through in the intrinsic or extrinsic pathway, there's not a whole lot of activated factor 8 or 5. And even when it's not activated, it'll still act as a cofactor, just not as efficiently or effectively. But when thrombin starts to become formed down here, it feeds back and not only activates platelets, but actually starts to activate those cofactors, both 8 and 5. And that really revs up its activity. So activated factor 8 can really enhance the activity of 9A and calcium combined. So it can enhance that complex and produce even more 10A. Thrombin can increase the activity of factor 5A, so the prothrombinase complex becomes much more active and can produce more thrombin. So ultimately, we lead to more and more 10A and more thrombin, which feeds back and just, again, through a positive feedback loop, enhances its own activity. Okay, so that's the review of the pathways, and that's sort of positive feedback loops that you can see with, say, 10A or thrombin, and how that can interact with, activating platelets and so on. And we know that a lot of those reactions take place on the platelet membrane as well. So platelets and the coagulation cascade do work hand in hand. Now, again, we want that all to occur at the site of the damaged endothelium. So we have to make sure that the normal endothelium doesn't have clotting going on, because we don't want to form clots where there isn't damage. And so we have several mechanisms in place to sort of keep our blood anticoagulated. and that relies on having normal, healthy endothelium. So as long as that endothelium is healthy, we can prevent clotting. As soon as we get unhealthy endothelium, damaged endothelium, then clotting cascades can occur, which we want if it's damaged, say due to some sort of injury. However, in certain conditions like atherosclerosis, which can cause endothelial damage and plaque in the arteries and so on, that can cause clotting because it's disrupting the endothelium. And so this is where they run the risk of things like strokes and heart attacks and so on. So let's look at the mechanisms that protect us from clotting when we're not supposed to. So some of those are going to be, the first one we have here is antithrombin 3. Antithrombin 3's job is to inactivate 10A and thrombin, which are two of the ones that I emphasize as being part of the positive feedback loop. It doesn't act, though, without heparin. So heparin is a required cofactor for antithrombin 3. Now, we actually naturally make heparin, and it's located on our endothelial cell membranes. And so in that case, we have the heparin available on the surface there. We have the antithrombin 3, which is produced by the liver in the plasma, so they can interact with each other regularly. Of course, you might have seen that we give heparin to anticoagulate somebody in the hospital, but that heparin would have no activity whatsoever if there's no antithrombin 3. So these two are required together, and they'll inactivate 10A and thrombin, which will help to prevent any coagulation from occurring. Now there's something called protein C, and the protein C is activated by a receptor referred to as a thrombomodulin receptor. And this is a thrombomodulin receptor that's located on the endothelial surface of healthy endothelium. And it's activated, this receptor is activated by thrombin. So thrombin activates thrombomodulin. So if you're forming a clot, let's say nearby, some of that thrombin could actually escape. Most of the thrombin, when the clot is forming at a damaged site, 90% of it is going to stay local and kind of get trapped in the mesh that's forming. But some of it does leak out. We don't want that extra thrombin that's leaking out to cause clotting on the healthy endothelium that surrounds it. So what happens is that thrombin gets released into the healthy endothelium area. It can interact with thrombomodulin on those healthy endothelial cells, activate it, and that's going to activate our protein C. It's going to go from an inactive to an active form. And protein C inactivates our cofactors 5 and 8. And 5 and 8 can really rev up the coagulation cascade, so that helps to kind of slow that process down. The prostaglandins, I've kind of already mentioned some already. We talked about thromboxane A2, that's a prostaglandin. This is synthesized by the platelets, as you know, to activate platelets and to cause vasoconstriction. Prostacyclin is another type of prostaglandin, and this is synthesized by the endothelium. So the healthy endothelial cells will produce prostacyclin and release that, and I showed that on an earlier diagram. And that causes vasodilation and inhibits platelet activation. So it actually does the exact opposite of thromboxane, so it prevents its action. Okay. And then we have plasminogen, and plasminogen is the inactive form, which gets converted into plasmin. And plasmin actually utilizes proteolysis to break down fibrin units. It can break down the fibrin units before or after the mesh has formed. Now this is an important distinction. Antithrombin 3, protein C, and these prostaglandins can all help prevent the coagulation cascade from occurring. But once fibrin mesh has formed, okay, the only thing that can actually break it down once it's formed is plasmin. So that's the only one that can actually break down a formed clot. And so the way that works is thrombin, once again. Thrombin actually stimulates endothelial cells to release tissue plasminogen activator. Tissue plasminogen activator gets released into the plasma, and what it'll do is it activates plasminogen in the presence of fibrin. So as long as fibrin is there and there's TPA, that can activate plasminogen and turn that into plasmin, and the plasmin can then actually break down any fibrin mesh that's been forming. And in fact, you may recognize the term TPA. It's something that's used if somebody, say, has a stroke or an MI due to a fixed clot, and they can see the clot, they may give TPA to break that clot down, because heparin, protein Cs, and prostaglandins and all that cannot break down that existing clot, but plasmin can. So this is the antithrombotic summary that you guys have in your notes. I'll just review some of the main points here. Here we have the inside of a blood vessel. You can see the lining endothelium here and the collagen, the subendothelial collagen on both sides. And you see, here's the site of injury. All right, so we can see a clot's forming. So we have a platelet plug. As you see here, here's the platelets. They're being activated by the presence of that negatively charged collagen that's there. And as they activate, they release their mediators to activate more platelets. At the same time, we're also going to be activating the coagulation pathway. And the result is going to be a production of thrombin, and that's going to help convert fibrinogen into fibrin. That's going to create that cross mesh to reinforce that platelet plug. Now, most of that thrombin will stay, as I mentioned, at the site of injury, so it doesn't really leak out, and it continues to activate other factors and so on, so that the clot gets more and more reinforced. However, some will escape into the healthy areas, and again, we don't want clots forming in the healthy areas because there's no damaged tissue there, but luckily the healthy endothelium will prevent that. So let's say some of the thrombin actually gets out of that zone there. So here's some of our thrombin here. All right. The thrombin itself can actually act on, as you can see, there's some arrows here to this right here, which are thrombomodulin receptor that I mentioned, that activates protein C. Protein C can deactivate or inactivate factors 5 and 8. All right, so thrombin was responsible for that. Thrombin also stimulates healthy endothelium to release TPA. All right. So what this means is that thrombin not only plays a role in the clotting cascade and reinforcing it by positive feedback and so on, but thrombin itself in the healthy endothelium actually acts to initiate antithrombotic mechanisms. All right. Again, the TPA, when that's released, will in the presence of fibrin, will activate plasminogen into plasmin, and the plasmin can break down the fibrin into fibrin fragments that won't clot. Okay. And then lastly, the last one I mentioned was that there's heparin on the endothelial surface that interacts with that antithrombin 3, which inhibits thrombin and as well as 10A. And what you're seeing here is that a healthy endothelium will also release prostacyclin. So PGI2 is a prostacyclin which inhibits platelet aggregation and helps to also vasodilate. All right, so those are all mechanisms that are built into the healthy endothelium to prevent unwanted clotting. So again, now we form the clot. What's sort of the fate of the clot? What's happening with that clot? So what happens is we get what we call clot retraction, and that occurs pretty quickly, usually within about 30 minutes. And what that means is that the platelets that are all grabbing a hold of each other due to these pseudopods that are sticking everywhere, and they're aggregated, they're going to start to actually contract and pull inward, and that helps to bring the whole platelet plug and make it really tight, so it's more leak-proof, if you will. And then the mesh that's forming over from the coagulation also helps to reinforce that, so it becomes very tight, and it pulls the end of the two broken endothelial pieces, let's say. It helps to bring the endothelium together as it retracts, so it holds onto the endothelial surface, right? It's adhered to it, and it pulls on that as well as it's retracting. And so that makes it a nice tight seal. At the same time, those platelets are going to release something called a platelet-derived growth factor. And this is necessary because when this is secreted, that's actually what we call a mitotic stimulant, and that's going to stimulate fibroblasts, which can lay down things like collagen and extracellular matrix, and smooth muscles as well. And it's going to help them to stimulate and regrow and repair those damaged vessels. So while that clot's there, it's stimulating regrowth and healing. And then finally, I don't want that clot to be there forever, right? Once the tissue is repaired, I want to break that clot down, so we have fibrinolysis. So to do that, of course, we need plasmin, because as I mentioned, that's the only one that can actually break down an existing clot. So when that breaks that down, we get this fibrin-dissolving enzyme that's going to break that down. Now the timing of this is such that the plasminogen is really getting released into that forming clot as it's forming. And you would think that it would start to break down the clot as we're trying to create it. But its release is very slow. The kinetics of its release are very, very slow. And so it's slow to activate. So it can take a couple of days for plasmin to reach a level where it can start to dissolve the existing clot. So that gives the clot formation plenty of time and the healing, and it allows that to happen. And then we'll dissolve and remove that clot. All right, so this all comes down to homeostasis, right? So both the platelet plug and fibrin clots are temporary and will eventually undergo retraction and dissolution, as I mentioned. All right, but if we have a disruption between coagulation and anticoagulation, right? Because we're trying to prevent clots in healthy endothelial or healthy vessels, but we're trying to have clots occur in the right spots and in the right amount. Okay, so since there are so many different players, so many different factors and platelets and intermediates and so on, there's just a multitude of ways that we can target this with drugs, or a multitude of ways in which there can be problems that arise because we have a lot of moving pieces here. So if we have, for example, too few platelets or poor platelet function, right, reduced number of factors, things like that, we can end up what we call in a hypocoagulated state or hypocoagulable state. And we're more prone to bleeding then because the things that are supposed to be interacting are not doing the job, or they're just not present. On the other hand, we can have the opposite. We can have too many platelets, or a deficiency of our antithrombotic mechanisms like antithrombin 3, or a deficiency of protein C. And this can actually lead to hypercoagulation. Somebody who's hypercoagulable, so they're more prone to clotting. All right, so there's various diseases where we might see this. In terms of inappropriate clotting, all right, there's a very famous gentleman by the name of Virchow, and right down here you can see I already boxed it in red, but I'll circle it for you anyway because I like shapes. Virchow's triad, all right, so this basically, his triad, these three things will make somebody more likely to form a clot inappropriately, to make them more hypercoagulable, if you will. So hypercoagulability itself is the first one. What that basically means is the person has a predisposing condition for this, maybe a genetic disorder where they have a factor, a factor mutation or something like that that's predisposing them to actually form clots. On the other hand, it might be hemodynamic changes. These are fairly common to see actually. This is where there's some change in their blood flow. So the blood has more stasis to it. It's pooling more, and say in the veins we get a lot of pooling, which is actually why I put the picture of the airplane there. They'll tell you that you should get up and walk around and stretch your legs every so often during long flights, because if you remain seated for too long, that can cause stasis of flow. That stasis of flow, what ends up happening is you have all these clotting factors and platelets that under normal conditions are moving at a decent rate and don't have a chance to really interact with each other all that much. But these factors do spontaneously activate. So for example, you could have a factor 12 or a factor 3 or factor 7 or whatever will spontaneously just activate in the plasma. And that happens, just a certain percentage of them will activate. But nothing usually comes of it because we have all these antithrombotic mechanisms and they're flowing by so fast and they don't really get a chance to really interact with each other. So when you have stasis of flow, like pooling in your veins for a long period of time, when they spontaneously activate, they can actually interact with one another. And that can actually start to form a clot. You can actually start to initiate some of the cascade, and you can form a clot in static blood flow. Turbulence can do the same thing, which is why something like a condition like atrial fibrillation, which causes the atria of the heart to kind of fibrillate. So it's not really pumping, it's just kind of shaking, if you will. That causes the blood in the atrium to just kind of pool and shake around a little bit, and that has been shown to promote clotting. And then finally, endothelial dysfunction. Remember, the healthy endothelium is the one that helps to prevent us from forming clots in the first place through all the antithrombotic mechanisms. However, in this case, if they're damaged, like due to atherosclerosis, which is what I mentioned earlier, due to like high cholesterol and so on, they can actually cause activation of the clotting cascade, particularly things like the intrinsic pathway, which I mentioned. So you can see that, here's the list here. I said, stasis of blood flow, like a DVT or deep venous thrombosis, and AFib are classic examples of that. Endothelial damage due to atherosclerosis, genetically or acquired deficiency in, say, an antithrombotic mechanism, like factor 5 line mutation is a common one. Thrombogenic surface, so somebody had a heart valve that was replaced. The valves of our heart are aligned with endothelium, which would be healthy endothelium to prevent clotting. By replacing it with a mechanical heart valve, there's no endothelium lining that, and that makes it more prone to coagulation. There is a difference between whether a clot forms in the vein or the artery. So over here to the right, a venous thrombus, they tend to be like larger and more loosely arranged, and they contain more fibrin and red blood cells. And again, the flow is usually very slow in the veins there. In an arterial thrombus, where pressures are typically higher, we end up with more platelets, fewer red blood cells, and fibrin. All right. So usually in a venous thrombus, we usually refer to it as like a red thrombus because it has red blood cells in it. And then a white thrombus is usually more arterial because it's fewer red blood cells and fibrin. But ultimately, the issue is the same. When you form a thrombus, it can either sit there and form the clot, which is the thrombus, and obstruct flow. And if it's in major circulatory pathways, it can cause a lot of damage to anything downstream of it. If it breaks off, if a thrombus moves, so it forms in one spot and then breaks away and it starts to move with the circulation, then clots somewhere downstream, that's referred to as an embolus. All right. So for example, somebody had a pulmonary embolus. That's typically because they had a thrombus that formed in a vein, often in their legs, and it broke and it traveled all the way to their lungs, where it wedged into the circulatory system there, and that can cause trouble with breathing. So here is examples of deep venous thrombosis, right? So here on the left you see this is the legs of one person. It's not two people's legs here, and you'll see the leg over here. It looks erythematous or red, right, in color. It looks swollen, and this is due to the fact that there's a clot in the venous system there that was large enough to disrupt flow returning back to the heart, and it causes swelling and extravasation of fluid there, and it can often be very, very painful. And you compare that to the other leg, which you see is, which would be their normal leg, if you will. In a hospital, there's a lot of stasis in flow, which is again one of Virchow's triads indicators here for hypercoagulability. So oftentimes, we'll put devices on the legs there, right? These sequential compression devices. And we put on the calves and the quadriceps, especially in people who are bedridden for prolonged periods of time. Because if they're not moving, there can be a lot of stasis there, and that's going to make them more prone to forming clots. So these are actually key in helping to prevent hypercoagulability in hospital patients who are bedridden for prolonged periods of time. And then finally, I did mention before, we have the intrinsic and extrinsic pathways, and you can see a cartoon down there which kind of just lists the pathways in shorthand. And like I said, physiologically speaking, these are typically both activated because they do have crosstalk, which I demonstrated in previous slides. But we'll use a lab test. We'll take what we call a PT and a PTT to measure the time it takes for the extrinsic pathway and the intrinsic pathway to form clots. So the PT is also referred to as the prothrombin time, we usually call it PT, and it measures the extrinsic pathway. And the PTT, as you probably guessed at this point, all right, is the partial thromboplastin time that measures the intrinsic pathway. All right, so extrinsic and intrinsic. The way I remember one versus the other is that the PTT has this extra T inside. Between the P and the T, there's an extra one in, so that's the intrinsic pathway. It's just a stupid way I remembered it, but I always remembered it, so maybe it'll work for you. So PTT measures the intrinsic pathway, the PT the extrinsic. Now, with the PT, again, that's going to measure factors like 3 and 7, for example. The intrinsic can measure, or can take note of the factors 12, 11, 9, and 8. Now remember, they both go to the common pathway. So the way we could use this diagnostically as an example is if we say the extrinsic pathway is prolonged, it's taking longer to bleed than it should. Then we take a look at so we were measuring that with the PT. So we look at the PT and it's prolonged. Then we look at the PTT and we see the PTT is normal. Well, that tells me it's probably the extrinsic pathway, something wrong with factor 3 or 7 that's causing this prolongation of clotting. On the other hand, if both the PT and the PTT are prolonged, remember they share a common pathway. So that might be suggestive that one of the factors in the common pathway may be abnormal. All right. So this is how we would kind of compare one to the other to see if we can figure out which arm of the coagulation cascades are being affected here. It helps us to narrow that down. But more often than not, we were using it because we usually have patients on medications. Many patients are on anticoagulants, and so we have to monitor how anticoagulated they are. So an example I gave previously with the vitamin K dependent ones, 2, 7, 9, and 10, we use Coumadin or warfarin, right? That's an anticoagulant. Now I want to monitor how much I anticoagulate them, so we can do it to a degree. We can reduce their hypercoagulability to certain degrees, depending on how much Coumadin we give them, and we have to monitor that. So we measure their PT. So we'll follow the PT along with that. Then the INR is actually a ratio that we use of PT. And so we'll get more into this a little bit later, but the INR typically for a regular, healthy adult would be around 1. All right. If I have somebody on warfarin, which is going to make them, more anticoagulated, so they're hypocoagulable, usually we'll do that if somebody has AFib or certain conditions. Then we're looking for a different INR range, maybe something around 2 or 3 instead. Finally, the PTT, which is the intrinsic pathway, we may use that to monitor the effect of heparin. All right, increasing antithrombin 3 activity is which heparin's function. So we will monitor heparin's activity and anticoagulation via the PTT. So in other words, if heparin, for example, if we are using heparin, we can monitor how much we're anticoagulating the person with the PTT and see how long the PTT has been prolonged, as we're essentially inhibiting the coagulation.